What if I told you that the missing link to curing a complex disease has actually already been discovered?
Right.
Like it's been uploaded to a server, it's proven in a lab, but no human has actually written a paper about it yet.
I mean, it sounds like complete science fiction,
Yeah, it really does.
but it is actually the reality of where biomedical data is right now.
Yeah.
We're generating raw experimental data at a pace that entirely outstrips our ability to write traditional peer-reviewed papers about it.
Which means the cure is just sitting there, completely disconnected.
Exactly. It's just sitting in a database.
And that is the exact problem we are exploring in today's deep dive. We are looking at a really groundbreaking paper titled OptimusKG, Unifying Biomedical Knowledge in a Modern Multimodal Graph.
Yeah, by 𝗏𝗏𝗏 et al.
Right. And we are going to unpack exactly how these researchers built this massive new digital map of human biology.
It's a massive undertaking.
It is. We have a lot of ground to cover today, but my promise to you, the listener, is that we are going to distill this entire universe of data down into a neat 15 to 17-minute audio overview.
And it's a critical topic, too, because of the architecture they've built here. It fundamentally changes how artificial intelligence can interface with biological data.
Oh, absolutely.
I mean, it basically bridges the gap between what we theoretically know and what we can actually use in practice.
Right. But, before we get to the architecture itself, we kind of have to talk about why the current ecosystem is failing us in the first place.
Yeah, setting the stage is important.
Exactly. So I was reading through the background and summary section, which lays out the current landscape of AI in medicine. And the core issue there is that AI models are entirely dependent on underlying knowledge graphs, or KGs, to actually understand biology.
Exactly. And the background and summary section makes it very clear that current knowledge graphs are they're deeply fragmented.
Yeah.
We have these incredibly powerful AI models being deployed for drug discovery and disease diagnosis, right?
Right.
But they're running on engines built from disjointed, non-standardized data.
It's like putting premium fuel in a broken engine.
That is exactly it. Current KGs lack standardized schemas. They don't have robust provenance, meaning you can't track a specific claim back to its original source.
Wow.
And they severely lack rich metadata. So you often just get a single flat snapshot of a biological relationship that becomes outdated almost immediately.
Okay, let's unpack this for a second, because looking at the current setup, it feels a bit like using a GPS map where none of the street signs match.
Oh, that's a good way to put it.
Yeah, like half the roads are outdated and there's just no way to know who actually drew the map.
Right.
But I do want to push back on this a little bit because, from the outside looking in, it seems like this should be a solved problem by now.
You would think so, yeah.
Right, like we have massive cloud computing power today. Why can't we just take all of this biomedical knowledge from these different labs and hospitals and just dump it into one master spreadsheet?
I hear that a lot.
Why is it so hard to just combine the spreadsheets?
Well, because biology doesn't fit neatly into a standard spreadsheet.
Yeah.
When you try to integrate highly structured resources like, say, a curated clinical database with unstructured or semi-structured documents, you run into an absolute harmonization nightmare.
A harmonization nightmare.
Yeah, because you are trying to connect data across completely different scales of existence.
What do you mean by scales of existence?
So you have the molecular scale, like protein interactions, right?
Yeah.
Then you have the anatomical scale, the clinical scale, and then environmental exposures on top of that.
Oh, I see. So they don't even share the same foundational variables.
Precisely. How do you seamlessly connect a cellular component to a complex human disease phenotype or an environmental toxin without losing the highly specific metadata of each?
You really can't.
No, you can't. If you just dump it all into a SQL database, the nuances, like what specific experimental assay was used or what the statistical confidence score was, those just get stripped away purely to make the columns match.
Wow. That makes sense. It's almost like trying to merge a map of ocean currents with a map of highway traffic.
Yeah.
They both show movement, sure, but the underlying rules are entirely different.
That's a great analogy.
So how did the researchers actually solve this harmonization nightmare? To understand the mechanics of that, we need to look at section methods, and specifically Figure 2, which outlines the pipeline they call the medallion data lake architecture.
Right. The medallion architecture is essentially the engine of OptimusKG. It's designed to logically organize incoming data in successive stages of increasing structure and quality. The framework is divided into 4 distinct layers: landing, bronze, silver, and gold.
So looking at Figure 2, the progression from raw data to the final graph, it reminds me of a massive United Nations translation floor.
Oh, interesting.
Yeah, you have all these different data sets arriving. They're speaking entirely different languages, and they have to go through multiple stages before they can communicate with each other.
Exactly.
Let's break that down, starting with the landing layer.
So in the landing layer, raw data arrives from highly heterogeneous sources. We're talking APIs, FTP servers, static ZIP files, JSON, SQL dumps.
Just everything.
everything. But the critical rule in this layer is that the data is ingested as immutable artifacts.
Meaning they don't touch or alter the original files at all?
Exactly. They keep the raw original formats. This is entirely about provenance.
Right.
By keeping the original file untouched in the landing layer, you preserve full traceability. If a researcher ever questions a connection in the final graph, they can trace it all the way back to the exact unaltered file that was downloaded on a specific date.
Okay, so the delegates have officially arrived at the UN building. Next up is the bronze layer. This looks like the first major transformation step, where they convert everything into standardized, machine-readable formats.
All right. In the bronze layer, the data is still raw in terms of its content, but the containers are standardized.
Okay.
They convert these varied files into columnar formats, primarily 𝗙𝗫 files.
And this is a massive technical detail that we really shouldn't gloss over. The shift to a columnar format like 𝗙𝗫 is a big deal for AI.
It's huge.
Because in a traditional row-based database, if an AI wants to scan for every instance of a specific gene expression, the system has to load every single entire row.
Yeah.
Every patient record, every metadata tag, just to read that one gene column.
Which requires an immense amount of memory and time.
Exactly. But with a columnar format, the data is stored by columns instead of rows.
Right.
So the AI can just pull the specific gene expression column and completely ignore the rest of the massive data set. It makes querying incredibly fast and efficient.
That is the exact computational advantage, yeah.
Yep.
So we have the data in a uniform, efficient container, but it still hasn't been harmonized semantically.
Right.
That happens in the silver layer.
This is the actual UN translation floor.
Exactly. The silver layer is where the heavy lifting occurs.
Yeah.
You have quality validation, schema alignment, and cross-source integration.
Okay.
The researchers constrain all the nodes and edges to a shared top-level schema. They use a framework called 𝗏𝗁𝗀, and they define the BioLink model as their upper ontology.
Let's define upper ontology in practice, because this is really the core of how the translation works.
Yeah.
An ontology is basically a master rulebook for how we name and classify things.
Right.
If you have a clinical data set from France, a molecular data set from Japan, and an environmental data set from Brazil, they might all refer to the exact same biological mechanism, but they use completely different identifiers.
Right, exactly. One database might use an alphanumeric code, another might use a full chemical name, and a third might use a legacy symbol. Without an upper ontology, your graph would create 3 different nodes for the exact same entity, entirely missing the connection between them.
But the BioLink model forces them all to use the exact same namespace conventions. If a database tries to introduce a concept that doesn't comply with the allowed master rulebook, the pipeline just flags it. It forces all those different scales, molecular, clinical, environmental, to speak one foundational language.
Which ensures consistency across the entire graph. And once everything is actually speaking the same language, the data moves to the gold layer.
The final step.
Yeah, this is the final, highly structured output. It produces OptimusKG as efficient Parquet files, with nodes and edges stored separately.
Okay.
The properties are encoded using native columnar types, including deeply nested structures that capture all that rich scientific metadata we talked about earlier.
So we understand how the factory works. The architecture solves the fragmentation and the provenance problems. But a factory is only as good as what it produces, right?
Very true.
We need to talk about the sheer scale of the graph they generated. But before we discuss the actual size and scope, I want to explicitly point you to Table 1 and Table 2, which lay out the final metrics of the gold layer.
Yeah, the numbers are substantial. The scale of
Yeah.
what they've integrated is difficult to conceptualize without actually looking at the raw count.
I mean, looking at Table 1 and Table 2, OptimusKG contains 190,531 individual nodes.
Wow.
And these nodes span 10 different entity types, like genes, diseases, drugs, anatomy, biological processes, exposures, and more.
It's massive.
And connecting those nodes are 21,813,816 edges, spanning 26 different relation types.
Yeah.
And it's not just basic point A to point B links either. The graph includes over 67 million property instances encoding 110 million values. It is a staggering topography of human biology.
It really is. But, numbers in a vacuum don't tell the whole story.
Right.
We have to look at the origins of those 21 million connections. Like, where did this data actually come from?
Okay.
And for that, we turn to Table 3 and Table 4.
Which detail the primary data sets and the ontologies use.
Right. If we connect this to the bigger picture, Table 3 and Table 4 show that the researchers integrated 65 highly respected heterogeneous data sets. Yes. This includes major repositories, like BG for gene expression data, DisGenet for disease-associated genes, DrugBank, the comparative toxicogenomics database, and open targets.
Are really the heavy hitters.
Exactly. They pulled from the most critical silos in modern biology.
And to make sure those 65 data sets all aligned on that silver layer translation floor we just talked about, Table 4 shows they harmonized everything using 18 distinct ontologies. They extended that BioLink upper ontology with specific rulebooks like the gene ontology, or Go, and the Uber anatomy ontology, known as Uberon.
By doing that, the effective ontological coverage of OptimusKG becomes the union of all the ontology trees present in those underlying primary data sources.
Right.
Is incredibly comprehensive.
Okay, I do have a major critical question here.
Sure.
The scale is impressive. 65 sources, 18 ontologies, nearly 22 million connections.
Yeah.
But with a pipeline that massive, automating the integration of 21.8 million edges, I mean, how on earth do we actually know if these connections reflect biological reality?
That's the big question.
Right. If a research hospital is going to use an AI model trained on this graph to recommend a drug pathway, we can't just cross our fingers and hope the medallion architecture didn't make a semantic error during harmonization.
No, you absolutely cannot. That is the defining vulnerability of any knowledge graph at this scale.
Yeah.
Manual verification of 21 million edges is physically impossible. You would need thousands of postdocs working for decades.
Nobody has time for that.
Exactly. So the researchers devised an evaluation method that is basically the ultimate AI polygraph test.
Which brings us to the technical validation section of the paper, and specifically Figure 4.
Right.
Before we get into what Figure 4 reveals, how exactly did they set up this polygraph test?
So in the technical validation section, they detail their use of a tool called 𝗶𝗄𝗴𝗃.
Okay.
This is a frontier multimodal AI agent designed specifically for deep scientific research. 𝗶𝗄𝗴𝗃 has the ability to retrieve information and synthesize evidence from a corpus of over 150 million research papers and patents.
150 million.
Yes. It acts as an autonomous literature review agent, and it has been shown to match human expert performance on complex scientific queries.
So they essentially unleashed an AI super researcher that has read every medical paper ever published and just asked it to verify the graph.
Exactly. They didn't test the whole graph, of course.
Right.
They took a carefully stratified sample of seed nodes, and they excluded the massive overly connected hubs and the tiny isolated outliers just to ensure they had a truly representative sample.
That makes sense.
From there, they sampled neighboring connections, creating a validation set of nearly 4,700 true edges from OptimusKG.
Okay.
They fed those edges into 𝗶𝗄𝗴𝗃 and asked it to find peer-reviewed scientific or clinical evidence in the published literature to actually back them up.
I was looking at the results in Figure 4, and the success rate is fascinating. 𝗶𝗄𝗴𝗃 actually found supporting literature evidence for 70.0% of the sampled true edges. And it wasn't just a binary yes or no verification either. The researchers broke down the level of support into categories, ranging from weak to moderate to strong and very strong evidence.
Which is a very important distinction.
Yeah.
In biology, one paper claiming a connection isn't always enough to establish a consensus.
Right. And Figure 4 shows that over 29% of the edges were classified as very strongly supported. That means the AI found more than 6 separate independent research papers confirming that specific biological connection.
But you know, whenever you use an AI to verify the work of an AI-built pipeline, you have to control for hallucination.
Oh, definitely.
Large language models are notorious for trying to please the user by generating plausible-sounding evidence that doesn't actually exist.
Right, the fake citations.
Exactly. How do we know 𝗶𝗄𝗴𝗃 didn't just invent papers to make OptimusKG look accurate?
That is where the control group comes in, and it's a crucial part of the validation. To prove the AI wasn't hallucinating, the researchers generated a set of false edges. They deliberately created biological connections between nodes that absolutely do not exist in OptimusKG, and they fed these fake relationships to 𝗶𝗄𝗴𝗃.
And what was the AI's response to the fake data?
83.4% of those false edges received absolutely no supporting evidence.
Wow.
The agent searched 150 million papers and basically returned an empty hand, confirming that the connections didn't exist in the literature. It proves the evaluation was highly specific and that the AI wasn't just rubber stamping the data.
It establishes profound trust in the 70% of edges that were verified.
It really does.
But you know, this brings us to the most interesting dilemma in the entire paper.
Okay, here's where it gets really interesting.
Yeah.
Because if 70% of the real edges are supported by published literature, that leaves an undeniable 30% of the edges in OptimusKG that 𝗶𝗄𝗴𝗃 could not find any evidence for.
Right.
Almost a third of the graph's connections are completely absent from the 150 million paper corpus.
Yeah.
Did the medallion architecture fail? Did the translation floor map things incorrectly?
It's the obvious conclusion to jump to, right? But we have to look closely at the data before assuming the graph is just wrong.
Okay.
Specifically, we need to examine Figure 4J. This figure breaks down that literature support by the specific type of edge.
Okay, so it shows us where the errors are.
It shows us exactly where the unsupported connections are concentrated, and they are heavily clustered in two specific areas: anatomy gene connections and disease gene connections.
Okay, so why is the AI struggling to find published papers specifically about those anatomy and disease genes?
Because those papers don't exist. They haven't been written yet.
Wait, really? This is the twist that fundamentally changes how we view this tool.
It is. OptimusKG is not just aggregating old textbooks and published journals. Right. It pulls a massive amount of its data from primary experimental resources and functional genomic screens. We mentioned open targets earlier.
Yeah.
Platforms like that ingest raw experimental data, like high-throughput disease target screens or raw gene expression arrays straight from a lab. That data is uploaded to a database long before any human researcher synthesizes those raw numbers into a formal peer-reviewed narrative text.
Oh, wow. So the biological connection is real. It was observed in an experimental assay, the data points were recorded, and it was fed into the bronze layer of the pipeline.
Yes.
But because nobody has actually sat down, drafted a manuscript, gone through peer review, and published a journal article about it, paper QA3 just can't find it.
Exactly. Paper QA3 is a literature search agent. It only knows what has been formally written down in the 150 million papers it has access to.
That makes so much sense.
If a connection exists purely as raw data in an experimental database, the AI can't read about it.
So that 30% gap isn't a failure rate of the graph at all, it's a leading indicator.
Yes.
The 30% gap proves that OptimusKG contains valid biomedical knowledge that literally precedes the published scientific literature.
The graph is charting territory that hasn't even been formally named by the scientific community yet.
That is wild.
It completely inverts the standard model of research. Normally, we read papers to find data. Here, the data is moving faster than the papers can be written.
It changes the utility of the graph completely. OptimusKG isn't just a map of the known world, it is an active frontier.
And because the researchers are distributing this graph in those highly efficient 𝗙𝗫 files we discussed, it is incredibly accessible.
Right.
Any research team at any university or pharmaceutical company can download OptimusKG and plug it directly into their own machine learning models or use it for knowledge-grounded retrieval.
That's amazing.
They don't have to spend years building their own medallion pipeline.
Yeah.
The Unify translation floor is already built.
Which brings us to the end of our journey today. We started by looking at the messy, fragmented state of biomedical data. We walked through the medallion architecture, from the immutable raw files in the landing layer, to the columnar parquet files in the bronze layer, to the strict Biolink ontology harmonization in the silver layer. We explored the staggering scale of 21 million edges, and we unpacked how a massive AI polygraph test revealed that the graph is actually ahead of published science.
It is a remarkable technical achievement that sets a completely new baseline for how we integrate complex biological data.
And looking at the time, I am very proud to say we have successfully unpacked this massive universe of data right on schedule.
We did.
Ending our audio overview perfectly within our promised 15 to 17-minute window.
We covered a tremendous amount of ground. But before we wrap up, the implications of that unwritten 30% leave us with a fascinating new dilemma to consider.
Go ahead.
We know that OptimusKG holds vast amounts of raw, unwritten experimental connections.
Right.
We also know that we now have multimodal AI agents capable of reasoning over this entire graph.
Yeah.
So imagine a scenario where an AI scans OptimusKG and connects a raw gene assay to a specific disease phenotype, a connection that no human researcher has ever hypothesized, peer-reviewed, or written a paper about.
Okay.
The AI suggests a compound, and it works. It cures the disease.
Wow. I mean, think about it.
The data was generated by one lab's machine, harmonized by a pipeline built by another team, and synthesized by an AI model. If an AI discovers a life-saving cure entirely from unwritten automated data points, who actually owns the patent for that cure?
That is a wild thought.
Which is a question the medical and legal communities are going to have to answer very soon.